Hi, my name is Chris.
Hi, my name is Cameron.
We are Group 2, and we are going to be minimizing plastic waste in our oceans.
We're going to be doing that using an enzyme that we're injecting into algae to break down the plastics.
And my name is Shelly, and I'll be mentioning the goals.
My name is Logan, and I'll be talking about the methods.
My name is Josh, and I'll be talking about the ethical considerations.
I'm Franco,
And I'm Yasi.
and we'll be summarizing our findings for this project.
Hi, my name is Chris. I am part of Group 2, and my group is focusing on minimizing plastic waste in our oceans. We chose this idea because not many people are focusing on the dangers of plastic pollution in our oceans and how it affects marine life. In fact, in the North Pacific Ocean between Hawaii and California, there is the Great Pacific Garbage Patch that contains non-biodegradable plastic debris that is the size of Texas. Also, one garbage truck of plastic is dumped into the ocean every minute of the day, and with this constantly occurring, there will eventually be more plastic debris in the ocean than fish by 2050. Our group has developed a possible remedy to this situation that can decrease the amount of plastic debris that is polluting the ocean and prevent the Great Pacific Garbage Patch from growing larger in size. Here is Cameron to talk about the specific bacterial, enzyme, and algae we will be using.
Our research team decided to incorporate the PETase enzyme into an algae, which will in turn produce the PETase gene necessary to dissolve these plastics. The algae, Phaeodactylum tricornutum, is the ideal model diatom that we will be incorporating the PETase enzyme into, due to its various applications within the biotechnology field. The PETase gene will be inserted into the plasmid of the algae using electroporation. This enzyme will then catalyze the hydrolysis of PET plastic into monomeric mono,2-hydroxyethyl terephthalate, or MHET. Phaeodactylum tricornutum can exhibit different morphotypes, change cell shape, and is stimulated by environmental conditions. It can grow in the absence of silicone and survive without silicified frustules. Because its frustules don't become smaller, this allows for a continuous culture without the need for sexual reproduction. It is only one of 3 diatoms to have its entire genome sequenced. Thus, it has been applied as a biofuel precursor and recombinant protein expression due to its biosynthetic capacity and high growth rates.
So first off, before I mention the goals, I want to note why it is important to reduce plastic waste in the ocean. The first reason being that the continuously growing rate of plastic pollution has become a global crisis. The accumulation of plastic waste has grown to the point where billions of pounds of plastics can be found in swirling connections that make up about 40% of the world's ocean surfaces. Not only has plastic waste polluted the oceans, it also has a direct and deadly effect on the marine life living in the domain. Plastic waste creates hazards the marine life is unequipped to handle. Plastic waste has impacted at least 267 species worldwide, including 86% of all sea turtle species, 44% of all seabird species, and 43% of all marine mammal species. The plastic waste mismanagement is condemning countless marine lives to death by the entanglement, ingestion, and suffocation of plastic. Thus, our goal is to genetically modify this algae to be able to produce the PETase gene in order to reduce the plastic waste in the ocean. By creating a large amount of microorganisms with the PETase gene, it will be released onto the Great Pacific Garbage Patch to accommodate for the continuously growing rate of plastic pollution. In addition to releasing it onto the Great Pacific Garbage Patch, we will release the algae near drains, filters, and grates that lead to the ocean to further reduce the plastic waste. Overall, the modification of this algae will become a more environmental and efficient alternative to reducing plastic waste.
Our method to produce an algal organism that produces the enzyme PETase begins by utilizing the plasmid from Ideonella sakaiensis called PET21B. This plasmid contains a bacterial promoter called LAC-B, which will be cut out using the restriction enzymes BGL2 and NDE1 in order to later be replaced by an algal promoter. The algal promoter will come from the plasmid FCPF and would be cut out using the restriction enzymes BSA1 and ICEUL. To ensure that this promoter has the proper sticky ends, it will be amplified through PCR using the sequences of the restriction enzymes from the PET21B plasmid. After PCR, the algal promoter will be introduced into the PET21B plasmid along with DNA ligase to form a new plasmid. This new plasmid will then be introduced into E. coli in order to obtain numerous samples. The plasmid contains the AMPR gene, allowing us to select for it using ampicillin. After selection and adequate amplification, the gene can finally be introduced to our algae through electroporation, resulting in an algal organism capable of producing PETase.
Moving on, now it's time to talk about the ethics of our experiment. First, I would like to talk about why our experiment would be important and how it could change the environment for the better. So every year, about 100 million tons of plastic are produced all over the world. Out of this 100 million, 25 million tons of non-degradable plastic gets accumulated into the environment. This plastic has led to harmful plastic pollution. When plastic waste has begun to negatively impact the natural environment, it kills plant life and poses dangers to local animals and even the human population. This is because, as plastic is created to last, it is nearly impossible to break down, and burning plastic can be incredibly toxic. When plastic is dumped into landfill sites, it does not decompose at a fast rate and can pollute the land or soil around its areas. It can interact with water to form hazardous chemicals, which in turn can degrade water quality. Ways that our experiment is ethical would be that it would reduce the amount of plastic waste in the environment without harming anyone or any wildlife. It would also lead to the reduction of polluted groundwater reservoirs, keeping our water quality a lot better than if it were to be polluted. Another reason that it's ethical would be that it reduces harm on marine ecosystems, killing less wildlife because of our human mistakes, such as polluting the environment by throwing plastic bags or bottles into the ocean. Lastly, it would also decrease the rate of pollution due to plastic. And it would decrease the money used to eliminate or recycle plastic. Ways that the experiment is unethical would be that the experiment would be pricey to execute, and if the experiment were to go wrong and fail, we just lose that money. It would go to nothing. Another reason why it'd be unethical would be that it can cause an outbreak, such as like illnesses or harm the human population. And another way would be like, if that were to happen, how would we take care of that? Because it's something we've never worked with, and it'd be in the ocean, which takes up most of the world. Another thing it could do it could start deteriorating other things that we weren't planning on deteriorating, such as boats or human materials that we have out in the middle of the ocean. And yeah, the most unethical thing would be that it can affect human health, such as causing illnesses which we wouldn't want. That's about it. But overall, if this experiment were to be successful, the benefits would outweigh the unethical points.
In conclusion, the ideal purpose of our experiment is to establish a more environmentally efficient approach towards reducing plastic waste in our oceans through biodegradation. Ultimately, we will be genetically modifying a single-cell algae known as Phaeodactylum tricornitum by isolating the PETase enzyme from the Idionella saccharinisis bacteria and introducing the enzyme into the algae's genome. Although we do not know the exact results of our hypothesis without performing the experiment, we can hope that the host algae will carry out the experiment and produce enough PETase enzyme so that it can break down plastic.
Assuming that the experiment goes as planned and that the algae takes up the PETase enzyme and functions correctly in the host, we can develop new methods of reducing plastic waste by introducing the modified algae in oceans and freshwater ecosystems all throughout the world, starting with the North Pacific Ocean, which is the location of the Great Pacific Garbage Patch. As mentioned earlier, the experiment will create an efficient alternative method of reducing plastic debris in our oceans all across the world. After releasing the modified algae in the ocean, we will focus on placing the algae in large vats that will go in large bodies of water, such as the ocean, lakes, and ponds where the algae can grow and degrade the plastic. However, there would be some errors in the procedure that could result in the experiment not being successful. For example, the enzyme in the algae may not properly break down the plastic in the same way that it does in the bacteria Idionella saccharinisis. Time also plays a role in the outcome of the experiment. According to research from other scientific papers, the degradation rate of plastic by PETase is unknown, and as a result, we are unsure of the time frame of the experiment.
We hope that our research will be a catalyst to reduce plastic pollution in all environmental ecosystems and the first step for other experiments also hoping to eliminate plastic waste and preserve the earth. Thank you.